There are a vast amount of three-dimensional structures seen on the molecular level, but one of the most impressive is the double helix displayed by DNA. While some of its most spectacular characteristics are those of its ability to code genetic information, its complex physical and chemical properties are currently being uncovered. Jacqueline K. Barton, a 61-year-old female chemist, has dedicated much of her life to exploring these properties. As a current professor at the California Institute of Technology, she has developed a research team named the Barton Group that is focused on continuing the study of DNA's properties. Barton's career began at Barnard College, where she graduated with high honors, earning the A.B. summa cum laude in 1974. After her undergraduate, she attended Columbia University and was awarded a Ph.D. in inorganic chemistry in 1978. From there, she became a professor at Hunter College from 1980 to 1982, then at Columbia University from 1983 to 1989, and finally at California Institute of Technology from 1989 until now, where she currently conducts her research. Most of the prior research that was done on DNA explored its role in genetics. Barton decided to focus on the structure of DNA and how its chemical and physical properties enable the DNA to carry out its function. This type of research hadn't been done before and has led to so many new discoveries thus far. Barton has earned a total of 18 awards and medals for her work, along with being elected to a variety of academies. She was a recipient of the prestigious MacArthur Foundation Fellowship in 1991, as well as earning the ACS Nichols Medal, the Wiesman Women in Science Award, and many others. The most recent award Barton earned was the prestigious National Medal of Science presented by President Barack Obama. She received this for her work in the discovery of a new property of the DNA helix, called long-range electron transfer, and also for showing that electron transfer depends upon the stacking of the base pairs in DNA. Barton's previous work led to the discovery of DNA-mediated charge transfer. However, currently, her team is using inorganic concepts to explore metal complexes as DNA probes. In this research, she used octahedral metal structures to probe the DNA. She then designed and studied novel complexes that are capable of specific interactions within target sites on the DNA. This is important because it uses a different approach than the organic complexes that were previously studied. Two specific types of metal compounds were used, ruthenium complexes and rhodium complexes. The rhodium complex cleaves the DNA backbone, and by tuning its shape, a series of these compounds was developed. These complexes use intercalation to recognize specific DNA sequences. Metal complexes that are capable of recognizing sites on DNA can be used in biological research as well, including cancer diagnostics and chemotherapeutics. Ruthenium complexes, on the other hand, luminesce upon binding to DNA. They have been used extensively to study the mechanisms of cellular uptake and are currently being studied as candidates for identifying specific cancer cells within the body. Jacqueline Barton has accomplished much in her career so far. She has had over 300 publications of her research and will continue to delve deeper into DNA and its properties. That should give you a brief introduction about Jacqueline Barton and her work and what she's done so far. The reason I initially chose her was because she did much of her work with DNA, and I've always been interested in the biological side of things. Another thing that kind of drew me to her work is that I really liked how she focused on the properties of DNA rather than just how it works in genetic function and with genes and that sort of thing. It was kind of nice to see the inorganic side of that. Another thing that I really liked, and the reason that I chose her, was that she combined inorganic concepts with DNA and with the biological aspect of DNA. To start my research on Jacqueline Barton, I started just online looking up basic information about her, her age, where she went to school, what she's doing now, that sort of stuff. After I had an idea of who she was and what her research was about, I moved on to her publications. I found those on various databases through Doan's Library Network, and I was able to read through and gain some knowledge on not only her specific findings, but also what those kind of all meant together. Um, and then I did find a really cool website that I thought was really helpful. It was one about her research that had been put together at California Technical Institute, and that was just really interesting because it had information about her and then also the research that her and the Barton group have done and then what they're currently working on. So that was a really good resource. I used that one a lot. I also found on that website her email address, so I have sent her an email and I have not received anything back from that yet, so if I do in the future, I'll definitely let you know. And then another thing that I did was continue to read through her research, but then I also found a short 2-minute video online that I watched that kind of summed up the research that she had done in the past. And then upon that, I continued to look up information about the awards that she has gotten, and from there I looked up what research she has done to achieve those awards. And that pretty much sums up how I did it. Some of the things that I found interesting while researching Jacqueline Barton were, first of all, that she's a female chemist, which when I usually think of a chemist, I think of a man, so it was kind of cool to see the female aspect of things. And another thing is that she has an entire research team at Caltech University dedicated to just her work, and that has expanded and included a lot of members over the years. I also found out that she had over 300 publications of her work, and I thought that was pretty impressive to have that many contributions to science. Another thing that I really liked was that she was able to incorporate inorganic chemistry concepts into studying biological processes. She used a lot of transition metal complexes and specifically intercalation to study the processes of DNA, such as proteins involved in some of the processes as well as cleavage of DNA. I read an article that showed repaired DNA proteins and how those could be initiated and kind of mediated by intercalation with transition metal compounds. And so that was one thing that really stuck out to me, especially after learning about these transition metal complexes in class and the process of intercalation. Another thing that I really liked that she did was she discovered the electron transfer of DNA, and so that was kind of cool that she discovered this whole new property that DNA had, and those are some of the things that just interested me in her work. I hope this video has given you a good idea of who Jacqueline Barton was and what type of research she has done. I think that her research is really important because she chose to delve into an aspect beyond just the genetic role of DNA, and that hadn't really been explored before. And her contributions while studying and conducting this research have been really big and are really starting to contribute to the expansion of advances in medical technology, especially with those with diagnosing cancer and catching the early stages of cancer. Overall, I think she's a great chemist, and I kind of hope that she actually gets back to my email at some point. But I think that's all I have for you guys, and I hope you enjoyed the video.